Hey friends, welcome back to this channel, and this is the series of endocrine system, and this is the third video on endocrine system. In our first video we have discussed about the crine system. In our second video we deep dive into autocrine and paracrine signaling system, and there is a separate video on prostaglandin, which is a good example of autocrine system. So if you haven't watched those videos, please go and watch these videos, and all the links of those videos are in the description box. So today we'll be discussing about the priming effect and the desensitization and downregulation thing. So before I click on this clickable points over there, let's just do a freestyle on this thing. In the endocrine system, two cells are always involved. One is the source cell. The first one is the source cell from where the hormones will be produced, and the second one is the the target cell. So this is the target cell, and how this target cell will act. They have specific receptors on which the secretion from the source cell, that means the hormones from the source cell, can come and bind. So the hormones will bind with these receptors and they will form the HR complex, that means hormone receptor complex. Now when the hormones are releasing in a very small amount from the source cell, what they will do is they will go and bind with these receptors, and they will increase or give a positive feedback on the target cell, so that more and more receptors will be produced and will be placed on the plasma membrane of the target cell, so that when in future, when huge amount of the secretions are coming, when the hormones level are increasing, then they can go and bind with all of these receptors and can have their own specific effect. So they will have their specific effect. So this is called the priming effect. That means one hormone is increasing. So there is a hormone is increasing the responsiveness of the target cell. That is priming effect. And how this thing is achieved? By priming. So they are achieved by priming of receptors, rather to say specific receptors, and placing them to plasma membrane. So let's say let's have an example. Now we in our future video will learn that the hypothalamus produce a specific hormone known as gonadotropin releasing hormone, and this gonadotropin releasing hormone act on the anterior pituitary. Okay, so this anterior pituitary cell must have receptors for this gonadotropin releasing hormone. Now when hypothalamus secrete very little amount of gonadotropin releasing hormone, they will go and bind with the receptors which are present at a basal level on the anterior pituitary, and in turn they will provide a positive feedback mechanism, so that more and more receptors are produced and are expressed on the anterior pituitary cells, so that when these gonadotropin releasing hormones are releasing more and more hormones, they will go and bind with this receptor and they can have their specific effect. And in this case, the specific effect is they will produce, the anterior pituitary will produce gonadotropins like LH and FSH and etc. So that is the priming effect. So now let's see what this thing say. So what's that? The priming effect. More receptors may be formed in the target cell in response to a particular hormones. I hope this is clear now. The second point is small amount of gonadotropin releasing hormone secreted by the hypothalamus, for example, increase the sensitivity of the anterior pituitary cell to further GnRH stimulation, right? This is the priming effect caused in large part by the upregulation of the receptors. In this process, increased number of receptor proteins for the hormone being primed and inserted into the plasma membrane. Subsequent stimulation by the GnRH thus cause a greater response to the anterior pituitary. Hope the freestyle helped, and hope you get that idea. Now the next thing is desensitization and downregulation. Again, let's get back to a freestyle thing. Desensitization. Before understanding desensitization, let's have an idea. Let's have a saying that with increasing, we often say that with increasing amount of drug consumption, our human body is getting resistant to drug. So yeah, we say that we are getting resistant to drug. So this resistant thing is same as desensitization. Okay, so what happened is, let's say we have a target cell, we have our target cell, and we have huge amount of hormones which can bind with the target cell. Okay, so let's say we have huge amount of hormones. We have a very active source cell. This source cell is very active, and we have huge amount of hormone, and also the priming effect worked, and it has produced huge amount of receptors. Their sensitivity has also increased. Sensitivity of the target cell has also increased, but now the hormones are the concentration of hormone is so much high, so high that all the receptors are now saturated with the hormones. Okay, and again the hormone concentration is increasing. So will this increased hormone concentration have any effect on this target cell? The answer is simple no, because all the receptors are already saturated with the hormones. So there will be no effect. In fact, there will be desensitization. That means no matter how much we increase the concentration of these hormones, there will be no effect on the target cell. Okay, so the graph will be like, let's say we have the concentration of hormone. This is the hormone concentration, and this is the response. So it will go like this, and it will go like this. So at this effect, desensitization has occurred. Okay, so this is desensitized. And what is downregulation? To to maintain the hormone receptor complex, and to maintain the normal homeostasis, this target cell need to get back to its original form, to its native form. It needs to get back to its native form, where there is no hormone response complex or hormone receptor complex present on this target cell, where the target cell have some free receptors. This kind of state it should go. So for that, the downregulation occur. There comes the downregulation. So downregulation is nothing where the hormone receptor complex is degraded, and the receptor is now clear. Receptor is emptied or clear, we can say clear, so that now next set of hormone can bind with this receptor. So that is downregulation. And to maintain this downregulation, some of our hormones, we have a pulsatile secretion. Pulsatile secretion means, let's say we have a condition. We have some stress or something. So we have some condition. There are release of specific hormones, and they have their effect, and this condition is cured. So their condition is cured, and then the hormone level or the hormone concentration will go down. This hormone concentration will go down. So that is the pulsatile secretion. So pulsatile secretion means there will be secretion of hormone, and then there will be a gap phase. Then there will be again secretion of hormone. Then there will be again a gap phase. Now this gap phase are the window periods where the downregulation is occurring. Okay, so hope that makes sense. Now let's see what this say. Prolonged exposure to high concentration of polypeptide hormones has been found to desensitize the target cell. In adipose cell, if adipose cells exposed to high concentration of insulin and testicular cells are exposed to high concentration of luteinizing hormone, this desensitization and downregulation occurred. Okay. Now, so these are the examples of desensitization. To prevent desensitization from occurring under normal conditions, many polypeptide and glycoprotein hormones are secreted in spurts rather than continuously. So that is called the pulsatile secretion. So that is for the priming effect and desensitization. Hope that makes sense. Hope you got the idea and hope this freestyle helped. So see you in the next video where we'll discuss about these pre-hormones, pro-hormones, hormone interaction, chemical interaction and all. So thank you. If this content was helpful, please make sure to like, share, comment and subscribe. See you in the next video. Bye.